morning. Today we are going to talk about proteomic analysis of sialoliths from calcified lipid and mixed groups as a source of potential biomarkers of deposit formation in the salivary glands. So we have seen the sialolith formation, what are the main components of a sialolith. Now they are going to analyze the proteins of the salivary gland stones, and they are going to find any biomarkers which are responsible for the identification of salivary stone formation. So this article was published in the journal Clinical Proteomics with an impact factor of 3.3 in the year 2023. The novelty is till date only one potential biomarker, which is mucin 8, which we saw in the seminar. Mucin 8 was tested for salivary stone disease. Well, this study used MS-based mass spectrometry-based proteomics, where performing only one experiment can detect many potential biomarkers. So the aim of the study was to detect the protein and bacterial composition of salivary gland stones by performing a proteomic analysis on a group of 20 stones from patients diagnosed with sialolithiasis. So they collected the salivary stones from patients, and then they did the analysis and found whether they were analyzing whether they were able to get any biomarkers for the salivary stone formation. So the methods included collecting 20 salivary stone samples, and this was conducted in the Medical University of Dams, and construction of a spectral library for the this is calcified, this is lipid and mixed sialoliths. The first step is they crushed the stones into powder in a mortar, and 50 gram of the powdered sialolith was the spectra was recorded in the information dependent format IDA for the protein fractions after separation in gel, that is SAID-ALF, which is digested according to the FAS. FA is nothing but printer-aided sample preparation. And the individual IDA spectra recorded for each sialolith sample studied in this work. What is SWAT analysis? It is nothing but sequential window acquisition of all theoretical mass spectra, which is a technique in mass spectrometry used for quantitative proteomics. It's a type of data independent acquisition (DIA) that allows for comprehensive and reproducible analysis of proteins and peptides. Unlike the data dependent acquisition, which selects specific ions for fragmentation, the SWAT MS fragments all ions within the defined mass ranges, creating a digital archive of all fragmented ions. And this enables the retrospective interrogation of the data and more accurate and reproducible quantification. So since we are using the salivary stones, we are going to find what are the mass spectrometric data we are arriving and comparing it with the already present data. And there was a chromatographic separation, which is a basic pH HPLC fractionation for cryptic peptides, which was performed in HPLC system. Next, the in-gel separation was done, which is the second approach for automated electrophoretic separation using SAT ELF automated system, which obtained fractions digested separately according to the FASP and green. And later, the LC-MS-MS analysis was done, which is a triple TOR 5600 plus mass spectrometer. So it was in the range of 100 to 1800 mass per charge. And the qualitative analysis and quantitative analysis were conducted. The results of MS analysis performed in IDA mode were analyzed in Peak Studio software, and the raw data generated during the MS analysis had to be converted to the WIP format, and this is the methodology by which they did the qualitative analysis. The quantitative analysis spectra, they found all the mass to charge ratios, and they compared with the already available values, and they did a analysis in that. And all these things were statistically analyzed, and enrichment data analysis were also done. And this is the workflow of the thing. They collected the sialolith, crushed it, and then they did the protein extraction. Protein digestion was done. Later there was a final cleanup and LCMS was done. Finally the data analysis. This is the workflow which was followed in this study. And here they wanted two things. One is the qualitative analysis and another one is the quantitative analysis. In the qualitative analysis, the number of identical proteins for both groups was obtained as 162 human proteins and 151 bacterial proteins. So they were looking for both human and bacterial proteins, where they found these many proteins. In bacterial proteins, the sample 3 and 11 had many, this cal group has 12 and the mixed group has 22 proteins which were identified, and the most bacterial protein was from Actinomyces, Fusobacterium, Neisseria and Proteomic. And in human proteins, calcified group, lipid and mixed group proteins were detected, which was 42, 50 and 78 proteins respectively in each of these type of stones. So the quantitative analysis did the calcified sialoliths, where 109 proteins were quantitatively analyzed, and they found this KRP group of family of proteins in that. And in the lip sialoliths, they found 84 statistically significant proteins, where they found this trans-cycling, MGP, LTF, and SMRP. In lip sialoliths, they found 140 statistically significant proteins. So these were the things which they were able to analyze. The limitations of this study include limited sample size, and they used fragmented samples, and there was a lack of longitudinal data, there was no follow-up, and there was no in vitro or in vivo models. Protein identification sensitivity was also a limitation, and the bioinformatic inference limitations were also there. So the future scope of this study would be a large multicentric study, temporal studies where there's a whether there is any recurrence or response to any therapy. That kind of study has to be done. And in vitro modeling, targeted therapy development has to be done. And they did not explore the fungal and viral contribution. So that has to be included in the next session. Next study, future studies. And salivary biomarker panels has to be improved. There should be a functional validation also. So in conclusion, the study analyzed the protein composition of salivary gland stones classified into 3 groups, and the classification based on spectroscopic methods supported by proteomic analysis verifies the classification of clinical samples in a good way. Qualitatively, common proteins of all these were found, and most such proteins were identified for the calcified type, and the mixed group was the least unique. And the results also indicate that neutrophil extracellular trap formation may be crucial for the forming this type of deposit. So the net forms, which I already discussed in the seminar, plays an important role in the formation of the salivary stones. And the study guidelines, this study had an ethical approval from the medical university, and the sample collections were done and they were stored in -80 C for analysis. The standardized protocol was for the FASP methods, and for the SWAT MS for proteomic analysis was followed. Quality control samples were analyzed in triplicate, so they did a triplicate analysis. Data analysis was used by Peak Studio Spin Cytoscape, and validated the statistical methods, which had a p-value less than 0.05. And data sharing, they had shared the data of raw proteomic data in the Proteomic Exchange Consortium. The stroke guidelines were not explicitly mentioned or followed in the study. Although it's an observational retrospective study of clinical samples, there is no structured reporting aligned with stroke checklist in this study. Thank you.